Hello, everyone. This is your fifth lecture in the series of six. We only have one more lecture until we start learning the muscles themselves and the functions of those muscles, what movements they control. You'll also be getting your study guide this week to prepare for the test over the muscular system. And who knows what that test will look like, whether it will be something online or if it will be something that we'll take at school if we get to go back in May. So just stay tuned for that information, I guess. So today we're going to cover some other aspects of muscle contraction. For example, how is the intensity, how do we control the intensity of muscle contraction? And also, where do we get the energy for the muscles to contract? So before we do that, I would like to go over some of the information we went over last week, just to review, because I know that was probably the most confusing or the most difficult part of this chapter, the series of events that happen when muscles contract. Hopefully, you found some good resources on the agenda. So this is the motor neuron. This would represent one motor unit. So we see, the cell body here. Here's the cell body, and all these dendrites are receiving information from the spinal cord, right? And it's sending that information along the axon, right? And this will be one muscle unit because we have one axon, one motor neuron, and all of these muscle fibers are being activated by this one motor neuron. So one motor neuron and all of the muscle fibers it stimulates would be one motor unit. And here, where we see the axonal endings or the axon terminals, those are where we see the neuromuscular junction, and that's where all the magic happens. Whoops. Here's a review of the neuromuscular junction. Remember, as that impulse, that nerve signal is coming down the axon, so it's coming down the axon, and as it comes down the axon, right, it's coming down, it's going to make the membrane of this neuron more permeable to calcium. So calcium is going to diffuse in to the axon, and the calcium is going to trigger the movement of these synaptic vesicles carrying the ACH, right? This is ACH, our neurotransmitter. It's going to stimulate those or trigger those to move towards the membrane at the axonal terminal and release the ACH across the synapse, right? Here's our gap, our synaptic cleft. Or synapse. It's going to release those across the synaptic cleft, and they're going to bind to their receptors. And that this area where we see lots of receptors for the neurotransmitter ACH is called the motor end plate. So this whole area here would be a motor end plate. Okay. So when those ACH molecules bind, they're going to change the permeability of the sarcolemma, which is the muscle fiber membrane. they're going to change the permeability of the sarcolemma to where it's going to be more permeable to sodium. And so this is the sodium ions here, right? Sodium is going to diffuse in, and that is going to create the action potential. That's going to create the action potential. And the action potential is just a depolarization of the sarcolemma. So you can see where outside becomes more negative. You see these little negative, but now, so the outside becomes more negative because the sodium is going to be rushing in. So all those positive sodium ions are no longer in the extracellular fluid. And that upsets the polarity across the sarcolemma and depolarizes it, causing the action potential. So once that action potential occurs, it's like all or nothing. So that's going to, once that action potential occurs, that's going to mean that the muscle is going to contract because it's reached a threshold stimulus. Now, as the sodium is rushing in, that's going to cause some of the potassium ions down here. Here's our potassium ions. So this is going to cause the potassium ions to rush out, or kind of leak out actually. So the potassium ions inside are moving out, right? And those potassium ions moving out are going to restore that polarity. So that action potential occurs, that depolarization occurs, and then we have a repolarization. We have a repolarization as the potassium leaks out because we're putting those positive ions back out now they're not sodium, but we're putting those potassium ions back out in the extracellular fluid, and that restores the polarity across the membrane. Okay? Now remember, that action potential moves across the sarcolemma, and it's going to move down into these invaginations of the sarcolemma and move down this T-tubule, right? The T-tubule is part of the triad. We have the terminal cisterna right here of the sarcoplasmic reticulum, and here's our T-tubule that runs right in the middle. And we know that the main function of the sarcoplasmic reticulum is to store and release calcium. And so when that action potential comes down, it's going to open those electrically chemical gated channels, and calcium is going to be released. So the action potential, I'm just going to put AP here, action potential moves down from the sarcolemma into the inner parts of the muscle fiber. And we can see that all of these myofibrils have the sarcoplasmic reticulum kind of wrapped around them. And that sarcoplasmic reticulum is what stores and releases calcium. The action potential makes the terminal cisterna more permeable. It opens up those electrically gated channels and upsets that polarity, opens those channels up, and releases the calcium. And remember, the calcium is what is going to cause, it's going to be the final trigger for muscle contraction. Okay, so Here is one of the that diagram that goes through all of the steps of muscle contraction. The only thing that it doesn't go over is how those ion concentrations are restored across that sarcolemma. So how do we get sodium back out into the extracellular fluid and potassium back in? Well, that's done with active transport of the using the sodium potassium pump. Okay, so the polarity, right? The repolarization. That's kind of tricky. So the repolarization occurs with the calcium leaking out, right? So the calcium leaking out restores the polarity, so it repolarizes the sarcolemma. However, we have to make sure that we have sodium back on the outside and potassium back on the inside in case that muscle wants to contract again, right? So the restoration of the ion concentrations occurs due to the sodium potassium pump. So as soon as the action potential moves across an area, we have the leakage of potassium out with that sodium coming in, and then right behind that so right behind the depolarization, we have the repolarization because of the movement of potassium, and then once that muscle has contracted, we have the sodium potassium pump restoring the ion concentrations, if that makes sense. Okay? All right, hopefully you were able to see that in one of the videos that I posted. So that calcium, remember, the calcium that's released by the sarcoplasmic reticulum, by the terminal cisterna of the sarcoplasmic reticulum, is going to bind to the troponin, and it's going to move that troponin out of the way, right? It's going to move the troponin out of the way so that the myosin heads can bind, forming cross bridges. And as soon as the myosin heads bind, they're going to pull those actin filaments towards the center of the sarcomere. And it's going to shorten every sarcomere of every myofibril in that muscle fiber, right? So lots of shortening going on. So that's why we say that calcium is the final trigger for muscle contraction, because muscles can't contract if those myosin binding sites are blocked by the troponin tropomyosin complex. All right. So moving on today, we're going to talk about the contraction and how we can how the intensity of the contraction is controlled. So you need to know that muscle fiber contraction is all or none. So if that action potential is generated, if that stimulus is strong enough to generate the action potential, that muscle fiber is going to contract, right? However, not all muscle fibers of a muscle are always stimulated, right? So we might have different combinations of muscle fibers contracting, different combinations of motor units being activated. And so the more motor units that are activated, the more motor units that are going to stimulate muscle fibers to contract, the more intense the muscle contraction is going to be. And we call those intensities and those different levels of intensity graded responses. Okay, so we have different degrees of skeletal muscle shortening based on how many motor units are going to be activated for a certain type of response. For example, you wouldn't want your, when you're grabbing a can of soda, you don't want a real intense response, and your brain knows that, and so the amount of motor units that are stimulated aren't going to be there. It's not going to be as many motor units stimulated to contract for that type of response as there would be if you were, holding on to a ski rope or something, trying to, water skiing or something like that. So, graded responses, different degrees of skeletal muscle shortening. So let's look at those different graded responses. So again, the intensity of contraction depends on two factors. So increasing the strength of the stimulus. So we're increasing the strength of the stimulus from our spinal cord, from the motor neuron, depending on what the response needs to be. That means that we're stimulating more motor units. And you can see the threshold must be met here in order for contraction. And then once the threshold is met, then, the more muscle or motor units that are activated are going to increase the tension in that muscle, increase the strength of contraction. So you can see down here how this corresponds to the amount of motor units that are excited. So for a very light stimulus, right, there's only maybe one motor unit stimulated. And then as we increase the number of motor units that are excited, we have more muscle fibers that are going to join in and contract for that response. And you can see we have, for the greatest amount of tension here. So this would be the greatest amount of tension. The greatest amount of contraction. We have lots of myofibrils in that muscle fiber excited. Okay. All right. So again, let's look at the difference here. So we have 4 different. And the other way we can control the strength of the contraction is increasing the frequency that those muscle fibers are being contracted. Okay, so we can increase the number of motor units, but we can also increase the frequency with which those motor units are being activated. And so this gives us the different how the frequency affects the strength of contraction. So this first one is just a single brief contraction of a single muscle fiber. And so this is just a twitch. We call this a muscle twitch. And so this is not a twitch like you get when you have a twitch in your eye or something. When you have a twitch in your eye or, usually it's your eye, you can feel a twitch in your facial nerves. Your nerve is being irritated by something. So that's a twitch, something you can't control. This type of muscle twitch is something you can control. So this is just a single brief contraction, and this is not representative of a normal muscle function. And then we have tetanus, when we increase the frequency, we don't give the muscle fiber enough time to relax all the way. So if you go back to that other slide, you can see where we have a contraction and a full relaxation. Here, we've increased the frequency so the muscle doesn't get to relax all the way. And you can see when we increase the frequency, every time that muscle contracts, it's going to contract at a higher tension. And that's called the summing of contractions. And so that's another way, again, increasing the frequency is going to increase the tension of the contraction. We can increase the frequency even more. You can see where we have even more reduced amount of rest between stimulations, right? Or between contractions. And this is called unfused or incomplete tetanus. Okay. The last slide was just tetanus. This is going to be unfused tetanus because our relaxation phase is getting very short here. And if we increase the frequency anymore, there's going to be no relaxation. And this is called fused tetanus, right? And this is a sustained muscle contraction. So we have a single contraction that gives us enough time for full relaxation, which is called a twitch. And then we have tetanus, where we see the summing of contractions, but there is, a resting phase in between, but we've increased the frequency of that stimulation. We've increased the frequency of that contraction, and it's summing. Every time it contracts at a little bit higher tension. So we're summing those contractions and increasing the tension on the muscle with each contraction. And then we have the unfused tetanus, right? Unfused tetanus, meaning we still have relaxation between, but very, very, very little relaxation. And then when there's no relaxation shown, we have something called fused tetanus, which is a sustained muscle contraction. Okay, so those are our graded responses. So again, 2 ways to control the intensity of muscle contraction. The first way is to control the amount of muscle fibers that are stimulated, right? So how many motor units are going to be activated? And for those motor units, how many muscle fibers are going to be stimulated by those motor units? We have motor units that stimulate lots of muscle fibers. We also have motor units that might stimulate only 1 or 2 muscle fibers. In our muscles that we use for fine motor skills, such as the muscles in our fingers, we have lots of very small motor units that maybe control only 1 or 2 or control the contraction of 1 or 2 muscle fibers. In large muscles that we use for like our for running and walking, we have much larger motor units. Okay, so now we're going to talk about the energy. Where do we get the energy for muscle contraction? And this will be probably a review for some of you that have biology. We're going to talk about the energy pathways that produce ATP. And you might remember, or not, that ATP stands for adenosine triphosphate, right? And this is the cell's primary energy currency. And to get and to produce ATP, we have to use a molecule called glucose, usually, right? Usually we use glucose. However, skeletal muscle cells are a special case. Skeletal muscle cells can also use another molecule for producing ATP. And basically, when we talk about ATP, we're talking about either the phosphorylation. I don't think it's on here, but we talk about phosphorylation, where we take ADP and we add a phosphate, right? So ATP triphosphate means we have an adenine, right? And then we have 3 phosphates. I'm sorry, PO4. PO4. So triphosphate. And these bonds, should do it like this. These bonds between the adenine and the phosphate are high energy bonds. So when they're broken, they release energy. And so if we take ATP and make it ADP plus phosphate, right? We're breaking off one of those phosphates. We're releasing energy. Okay, that's called hydrolysis. So hydrolysis is the releasing of energy by breaking off a phosphate. And then if we're going to add a phosphate to make ATP, this is phosphorylation. All right? So phosphorylation and hydrolysis. So let's look at, I'm going to erase this because this first energy pathway really has nothing to do with glucose, just ATP. So I should have probably left ATP on there. So you have that ATP is adenosine triphosphate. Right? Okay. So the first energy pathway that muscles can use is something called the direct phosphorylation of ADP by a reaction with CP. So CP is creatine phosphate, and I'm sure you've all heard of creatine, right? People that want to build muscles take creatine. But just taking creatine is not going to build muscles. You also have to work out. What creatine does, it's one of those extra, it's kind of an extra. So it's going to provide you another pathway for ATP production. So you can work out even longer. And by working out longer, you'll be able to, theoretically build muscles quicker, right? Because you'll have more energy available. So CP is a high energy molecule. It's only used after ATP is depleted. We don't have any oxygen left to use any of the other energy pathways. But creatine phosphate, even though muscle fibers have 5 times the amount of creatine phosphate as other body cells, the supplies get exhausted very quickly about 20 seconds is all you have with the creatine phosphate that occurs naturally within your muscle fibers. So this is a really easy kind of reaction here. We just take the phosphate off the creatine and add it to ADP to make ATP, right? It's important to know the ratio of ATPs we get per creatine phosphate. And so there's 1 CP equals 1 ATP, right? Which makes sense because there's 1 phosphate molecule on every creatine phosphate. So we add that to ADP, get ATP. And again, those stores don't last very long. Okay, so the direct phosphorylation, which means adding, right? Phosphorylation means adding a phosphate to ADP. All right, so that's the first energy pathway. And then this next one, this one is the one that you'll probably remember from biology. This is aerobic respiration. And aerobic respiration, the first part of aerobic respiration, both aerobic and anaerobic start out with glycolysis. And glycolysis occurs in the cytoplasm. Okay? Now, if oxygen is available, that the aerobic respiration, one of the molecules from glycolysis, if there's oxygen available, one of those molecules is going to move into the matrix of the mitochondria, and with oxygen, it's able to make lots and lots of ATP. If there's no oxygen available, we have to just stick with anaerobic, and we'll get to that in a minute. So let's look at aerobic respiration. We call this a metabolic pathway. It's actually a catabolic reaction. Remember, catabolic means to break down a molecule, and the molecule we're breaking down is a 6-carbon sugar called glucose. 1, 2, 3, 4, 5, 6. This is glucose, and it's going to be broken down first into 2 3-carbon molecule, or 3-carbon, 2 3-carbon molecules called pyruvate. Pyruvate, or pyruvic acid, right? And to do this, in order to break that down, we have to start out using 2 ATPs. So we have to put energy in order to get energy out. So we have to use 2 ATPs to break down the glucose, right? There's an intermediate step here where we also are going to be able to produce 2 ATPs. And actually we produce 4 ATPs. So we produce 2 per pyruvate molecule. So we have this intermediate step here, which I'm not going to go into. This isn't biology, but we have to put 2 in. We actually make 4. So we have a total of, or a net of 2 ATPs produced in glycolysis. And then, like I said, if oxygen is available, these 2 pyruvate are going to be further broken down into 2 2-carbon molecules called acetyl-CoA. And acetyl-CoA is what moves into the matrix of the mitochondria. And we go through the first one is the citric acid cycle. Let's remember the citric acid cycle. citric acid cycle is where CO2 is produced, and also two more ATPs, right? Because we have to turn twice. The citric acid cycle has to turn twice for each glucose molecule. And then the high energy electrons, there's a whole, we also use energy electron carriers in the citric acid cycle. And those high energy electrons are transported to something called the electron transport chain, which also occurs in the matrix, but also utilizes the inner membrane of, like in here, remember, this would be the inner membrane of the mitochondria. So that electron transport chain produces 32 ATPs with those high energy electrons. We won't go into the chemiosmosis and all the different steps of that, but altogether, aerobic respiration produces 36 ATPs. So, and this is also, the electron transport chain is where the water is produced. So you can see, remember, respiration is glucose, C6H12O6, plus oxygen produces carbon dioxide and water. That's what we respirate, right? That's what the cells bring back from our blood. The waste products are brought back to our lungs. We breathe out carbon dioxide and water, and then ATP, right? Make ATP. So here's our starting glucose, our 6-carbon glucose, our oxygen. We need oxygen in order for this to occur, right? But we produce, in the citric acid cycle, we produce the CO2. And in the electron transport chain, we produce the H2O. And in the process, produce 36 ATPs. So this is a slower reaction because of all the different steps it has to go through. However, it produces the highest amount of ATP, right? So one glucose molecule, one C6H12O6, produces 36. So we have 1 to 36 in this energy pathway. So aerobic respiration is the most efficient. Now, muscle fibers, however, you use up the oxygen very quickly. We do have a special protein that helps to grab oxygen. It's called myoglobin. It's one of the enzymes, proteins that helps to attach oxygen to kind of grabs the oxygen for the mitochondria. However, muscle fibers run out of oxygen very quickly and have to use an anaerobic pathway. So let's look at the anaerobic. Now, we do, aerobic respiration occurs first, right? And it provides, depending on the intensity of the exercise or the activity, it can last for hours doing regular activity. However, if you're sprinting, if you're, lifting heavy weights, for a muscular endurance or muscular strength training, those oxygen stores are going to be used up much more quickly. And so we're going to look at what happens when we don't have oxygen available. When we don't have oxygen available, we use the anaerobic pathway. And so anaerobic means, right, without oxygen. Now, I want to make sure that you understand that glycolysis is the first step in both pathways. Okay? So, at the end of glycolysis, when we have the pyruvate, if there is no oxygen available, the pyruvate can't be broken down into the acetyl-CoA because oxygen must be available. Oxygen is the final electron acceptor in the electron transport chain. So without oxygen, we can't move into the citric acid cycle or the electron transport chain. So instead, there's got to be some way to regenerate that NAD, the electron carriers and everything. So anyway, if we don't have oxygen available, the pyruvic acid, right, it doesn't break down. So we start out with the 6-carbon glucose, right? And it is broken down, right? We have to input our 2 ATP molecules. It's broken down into the 3-carbon, 2 3-carbon pyruvic acid molecules, right? That's this right here. We don't need oxygen for that, right? We just need ATP to break it down. Instead of being broken down into the 2 acetyl-CoA and moving into the, this is still happening out there in the cytoplasm. Instead of breaking down these 2 pyruvic acid molecules into 2 2-carbon acetyl-CoAs, we're going to convert this to lactic acid. So lactic acid is just an intermediate organic molecule that most animals, like us, use in anaerobic respiration. Okay, so the lactic acid is used, and then the lactic acid is transported to the kidneys actually, and is further broken down, and then we can use the components of lactic acid for energy pathways. All right, so we have to input 2 ATPs. Breaking this down, we have the electron carriers that also produce 4 ATPs. Okay, and the 4 ATPs, we produce 4, and so we have 2 net ATPs from glycolysis. So anaerobic respiration or anaerobic glycolysis, we get 1 glucose equals 2 ATPs. So you can see that this is, now this is a much quicker process, right? It's much faster. However, it's definitely not as efficient. And it only gives you about 30 to 60 seconds of energy. Okay? Okay. So that's all I have for you. Again, we have one more of these lectures, and be looking for that study guide that you can start working on in your agenda. Okay. All right. Well, have a great week. If you need anything, I will be in the office hours from 1 to 2 on Tuesdays and Thursdays. All right. Bye.